Hello students, welcome to the third lecture of first week. We were discussing many of the components we have already discussed in our first and second lecture, and in this third lecture let's move on to the other components. Now respiration, as we know, that is the process through which energy rich molecules are broken down to release energy from these molecules and utilize it for the cellular and metabolic process. Now this mitochondria is found in plant cells also, and their numbers can vary from few to few hundreds depending on the requirement of a plant cell. Now generally the shape of this mitochondria is cylindrical and elongated, and the length can vary from 1 to 4 micron and the diameter generally 0.2 to 1 micron, an average of 0.5 micron, and this is the site of ATP production. Okay, and it also produces many other carbon skeletons which could be used for other metabolic process in the cytoplasm. Now when we look at the structure of mitochondria, we see that it is also a double membrane structure. It has outer membrane and inner membrane, and inner membrane we can see that it is present in the form of foldings which is known as cristae. Now this cristae or the foldings actually increases the surface area of the inner membrane, which are site of various processes that undergoes in mitochondria. Now inside this inner membrane you have a matrix which also contain various metabolites, various enzyme proteins which are responsible for many other processes which is performed in the mitochondria. This mitochondria also contain its own DNA and ribosomes, so it can also perform some of its functions responsible for synthesis of protein enzymes. It is also considered a semi-autonomous body because it contains its DNA, it can synthesize its RNA and protein, and the origin of this mitochondria is also believed to be from ancient symbiotic association of an ancient independent cell, and then which is engulfed by a eukaryotic cell, and after that they started behaving as a single cell, but many of its genetic material were lost, and many of the things responsible in mitochondria is synthesized by the nuclear DNA. Now in mitochondria also we can, if we try to analyze the structure of this mitochondria and the membranes of the mitochondria, we can understand that how the structure is related to its function. Now in this case also, as we have studied in chloroplast, if we observe this mitochondria, the inner membranes which have lot of foldings, okay. Now if we elaborate these inner membrane, it is also made up of phospholipid bilayer. So two bilayers are there. Similarly in this case also we can observe these phospholipids, okay, and in this also lot of protein complexes are there, okay, are embedded in this, which are responsible for the process related to aerobic respiration. Now when we talk about aerobic respiration, we talk about various three types of process which takes place in aerobic respiration, that is glycolysis, Krebs cycle and electron transport chain. And electron transport chain. So glycolysis takes place in cytosol and Krebs cycle takes place in the matrix of this mitochondria, and electron transport chain takes place in the inner mitochondrial membrane, and all these processes together will help in complete breakdown of a molecule to release energy which is stored in these molecules through the process of finally to the process of photosynthesis. Now, in this case also, the electrons will be transferred from one molecule to another molecule, and because of to increase the efficiency of all these processes, the foldings are there, and finally at the end, energy will be released in the form of ATP. Okay, and it is known as aerobic respiration because at the end, oxygen will be the terminal electron acceptor. So, oxygen is required for this process, and therefore, it is known as aerobic respiration or oxidative phosphorylation. Now, coming to the next important organelle, it is the endoplasmic reticulum. Now, endoplasmic reticulum is a interconnected membrane system which is distributed in the cytoplasm. Now, this membrane system is made up of sac-like structure, flat plate-like sacs which are known as cisternae. Okay, it is made up of tube-like structures which are tubules and small rounded vesicles. So, they all form the endoplasmic reticulum. Now, it is continuous with the plasma membrane, Golgi complex and nuclear membrane. So, when you observe, suppose this is the nuclear membrane, okay, this is the nuclear membrane. So, this membrane will be continuous with the endoplasmic membrane. And finally, it will also be connected with the plasma membrane. Okay. So, it forms an endomembrane system, the membrane which is inside the cell, okay, distributed in the cytoplasm. Now, it is made up of single membrane and a space is there between this membrane where a lot of metabolic processes occur. So, here you can see flat membranous structure, and each membranous structure, flat structure, is known as cisternae. Basically, now the function of this endoplasmic reticulum in synthesis of protein as well as synthesis of lipid. These are divided into two types. We will have smooth endoplasmic reticulum, okay, and rough endoplasmic reticulum. It is called as rough endoplasmic reticulum because it contains a lot of ribosome present on the membranes, on the outer wall of this membrane of endoplasmic reticulum, giving them a rough appearance, that and because of that it is known as rough endoplasmic reticulum. These ribosomes are involved in protein synthesis. This protein is synthesized and it is transported inside the lumen of this endoplasmic reticulum, where it is modified, it is packaged, and finally it is transported to Golgi bodies or for further packaging and secretion. Then you have smooth endoplasmic reticulum, which does not have ribosome, and it is basically the next one is smooth endoplasmic reticulum, which does not have ribosome. That's why it is known as smooth endoplasmic reticulum, and it is involved in lipid synthesis basically and detoxification, like waste substances or the toxic substances which are harmful, they enter into the smooth endoplasmic reticulum and they are modified. So, it is detoxificated. Generally, it is found in those cells which are involved in lipid synthesis, basically like oil seeds, nectaries and glands. Smooth endoplasmic reticulum will be present. Now, in rough endoplasmic reticulum, we have seen presence of ribosomes. Now, ribosomes are the site of protein synthesis. Basically, they are not bounded by any membrane and made up of ribosomal RNA and protein. And they have two subunits, okay, a larger subunit and a smaller subunit. Now, these ribosomes differ in types which they are present in prokaryotic cell as well as eukaryotic cell, and in prokaryotic also mitochondria and chloroplast contains 70S type. This 70S type subunits are further divided into 30S and 50S type. Now S is the sedimentation coefficient. Okay. Eukaryotic cells means in eukaryotic cell also, apart from the mitochondria and chloroplast, the one which are found in the cytoplasm as well as studied in the endoplasmic reticulum, 80S type of ribosome is found, which has larger subunit of 50S and smaller subunit of 40S. Now, this ribosome actually synthesizes protein with the help of mRNA. Okay, other mRNA and tRNA, they also participate. So, this mRNA comes and gets bound by these two subunits of ribosome. Many a times it has been seen that one single mRNA could be bound by various ribosome, and this type of structure is known as polysome. And this type of structure is also visible under electron microscope and giving rise to multiple proteins. That means this increases actually the efficiency of protein synthesis. The next important organelle is Golgi apparatus. Now, Golgi apparatus consists of dictyosomes and vesicles. Basically, they are stacks of flat membrane bound cavities. Now, in animals actually this Golgi apparatus is slightly less simpler, and it is more complex in plant system, where several groups of these stack-like membranous cavities are present, giving rise to a complete Golgi apparatus, and each those stacks are known as dictyosomes. So, these dictyosomes basically they individually contain several stacks of flat membrane cavities, and basically 5 to 7 membrane cavities are present, and each one known as cisternae. You can also see lot of small vesicles also which are attached with this Golgi complex. So, basically it is also similar to that of endoplasmic reticulum, but you will not find connectivity between the cisternae, they are individual stacks. Now, this dictyosome, they exhibit polarity. The one which is receiving vesicle, the incoming vesicle comes from the endoplasmic reticulum, and this is the cis phase. Now, these vesicles, they enter into the Golgi bodies, they are further modified. Some carbohydrates groups are added, proteins are modified. So, many things of modification and packaging undergoes in this Golgi complex, and then finally they are secreted out from the other end, and this end is known as transphase. And many of the vesicles which are coming out could be secreted outside or could be part of various other cellular components of a cell. Now, this dictyosome help in plate formation during cell division, that many a time cell wall component is required during cell division. So, these vesicles come out and they get attached at the plate layer from the Golgi complex. Similarly, the non-cellulose polysaccharide which are present in cell wall as well as in the form of slime at the root cap tip, they are also secreted from these Golgi complex. Many a times the carnivorous plant secrete enzyme which are actually formed through Golgi apparatus. Now, the next very important component of plant or any living organism or any living eukaryotic organism is nucleus. Nucleus is a very big organelle. As we can see them under light microscope, we can stain them with a particular procedure is there, we can stain them and it could be easily visible under light microscope. It is a rounded structure. We know that nucleus is the site for storage of genetic information. And this information is present in the form of DNA. So, that DNA is present in the nucleus which controls all the metabolic process of a cell or of an organism. Now, generally one nucleus is found in plant, but many times more than one nucleus could also be present. Now, multinucleate condition could also be visible in other organism, like in fungus also you can find multinucleate situation. In plant also In tapetum, that we have studied before, that tapetum is the outer wall surrounding the microsporangia where pollen grains are present. So in this tapetum also, lot of multinucleate, lot of nucleus is present. It also exhibits multinucleate condition. This nucleus is bound by a double membrane envelope, as in case of chloroplast, as in case of mitochondria. So it will also have an outer membrane and it will also have an inner membrane. In between you have an intermembrane space is there. Okay, and in that membrane lot of pores we can find in the nucleus. These pores regulates the trafficking of molecules into and out of the nucleus, and these pores are also basically made up of protein complexes that selectively guide the entry and exit of metabolites. Inside the nucleus you will contain a fluid filled matrix which is known as nucleoplasm. Along with that, you will also see a structure, a subnuclear structure which is known as nucleolus. This nucleolus is actually the site of synthesis of ribosome. It contains rRNA, ribosomal RNA and ribosomes. So ribosomes are assembled here and they require protein and they require rRNA. So this nucleolus is attached to that part of the DNA where rRNA genes are there. They are transcribing rRNA genes, and the ribosome subunits enter into the nucleus through the pores from the cytoplasm and they are assembled there. Along with that, you can see lot of thread-like structures. Basically in interphase nucleus, lot of thread-like structures you can see which is known as chromatin, and these thread-like structures are actually DNA which is bound with protein. At some points you can see that this thread is highly coiled, okay, it is more condensed, that is the heterochromatin region, and at some places this is loosely packed, so it is euchromatin, and we can say that when it is loosely packed, the DNA is more active because when the threads are more exposed, the DNA will be more transcribed. The process can take place with more ease, and when it is packed, so definitely this synthesis will not occur. Now this DNA, so we have also heard of a structure known as chromosome. So when this DNA has to enter a division phase, these chromatin, they condense themselves and pack themselves in the form of chromosomes. So basically chromosomes are condensed chromatin, and lot of proteins helps them in condensation, and this they do so when they have to divide. So you would have seen cell division, I think so you all learned cell division, basically the process of mitosis, although meiosis is also there, but that takes place in sex cells, whereas mitosis takes place in all the somatic cells. So during chromosome division, the cell will align at equator and then they will divide. So all these stages, and finally they will give rise to two cells having equally divided chromosomes. So this all structure also you will see in a dividing cell. Now the next important component of a plant cell is presence of cytoskeleton. Now cytoskeleton is a network of protein strands or fibers which are found in the cytoplasm, and these structures are essential for the maintenance of plant structure and function. These are, you can say, these are network of thread-like structures which are distributed in the cytoplasm, and they help in maintaining the structure of a plant cell. These cytoskeleton are composed of microfilaments and microtubules as well as associated proteins. Another important cytoskeleton is also there, intermediate filaments, but that is generally not found in plants. The plants are basically contain microfilament as well as microtubules. Now, microtubules are basically bigger, they are rod-like structures, they are bigger or you can say broader than microfilaments. Each microtubule is made up of two types of globular protein. Alpha and beta, and these two globular proteins form a dimer. Okay, they associate in the form of a dimer, and they form a helical structure. And when they form, you can see here the formation of a helical structure of these dimer. And if we observe them under transverse section, we can see here the distribution of these dimers, and as many as 13 subunits we can see in this cross section. The diameter of this cross section is approximately 25 nanometers, so it basically forms a rod-like structure. Talking about microfilaments, they are polymers of protein actin. Okay, so actin proteins, they join together to form a single thread-like structure, and two thread-like structures, they wrap around each other to form a slightly thicker rope-like structure. Okay, along with these microfilaments and microtubules, certain motor proteins are there which help in moving the vesicles and organelles through this fibrous scaffold. These microtubules and microfilaments, they are involved in motility. The movement of the cell is controlled by these fibers. Then they are used in intracellular transport, like suppose a vesicle has to move from Golgi apparatus suppose to cell membrane, or any other organelle, suppose mitochondria has to move from one place to another. So they take help of these microtubules, and following the path of the microtubule and with the help of motor proteins, they get attached and they are moved on from one part to the another part. They also help in cell division, because during cell division, microtubules bind the chromosomes. We have seen during cell division, the chromosomes bind at the centromere of these microtubules, and they pull the chromosomes apart. So they help in cell division, they help in endocytosis. Okay, when engulfing, that is engulfing some food particles or all those processes, but this is not applicable in many of the plant cells. They also provide support to the internal organelles, to the cell, and they anchor the cell components at their location. Many of the cellulose fibrils which are found in the cell wall, they are also located at their position because of the presence of these cytoskeleton, and they do so actually because they can easily polymerize and depolymerize to change its shape. They are not actually fixed depending on the requirement. Suppose they have to move, they have to transport, they have to pull something, so they can polymerize and depolymerize. Now, this was all about the third lecture, and with this we end up with our topic of plant cell structure, and I hope you all would have got the basic understanding of a typical plant cell, the important organelles which are present in a plant cell, and how they perform a particular function for the overall process, for the overall development of an organism. Now at the end, I would like you all to try to find out the answers of few questions. First of all, there is a picture that I've already shown you, that with the help of some staining procedure, which is very simple, and I think so you all would have studied in your lower classes, that there is a staining procedure, a particular plant cell is used, and to show the stages of different mitotic division and to identify the nucleus. So I want you all to identify and to find out this staining technique. You find out the stain which is used and the organelles or the cell which is used to prepare the slide for identification of nucleus. And then in this figure, I want you all to identify the stages of cell division. Particularly, these are all stages of mitosis which occurs in somatic cell. So try to identify these stages. So thank you for this lecture, and we will meet again in the next one with another new topic.